Okay, it just hit 1:32 p.m. UK time on my watch. Uh, Hello, everyone, and thank you so much for joining us, and welcome to the third iteration, or rather edition, of the GSU Malaria General Club Monthly Series for 2025. Today's webinar is a vector-focused one, which is going to be referencing our speaker, Brian Polo's recent preprint titled Genomic Surveillance Reveals Geographical Heterogeneity and Differences in Known and Novel Insecticide-Resistant Mechanisms in Anopheles arabiensis across Kenya. My name is Polo Chauke, and I'm a training coordinator at the Genomic Surveillance Unit, working with a team of awesome people, some of whom are in the room right now, to organize these monthly events. Just a few ground rules. The General Club is going to be recorded, or is being recorded, and is going to be available on our Malaria Gen YouTube channel later today, so you can rewatch the presentation or also share with your colleagues and wider network as well for those of your friends and networks that couldn't attend today. Kindly stay on mute so that we don't disturb our speaker. We will have a Q&A session at the end where you can either raise your hand and we'll allow you to speak, or you can type your question as well as comments in the chat or Q&A box, and I'll read them out for Brian to respond to. Without further ado, I would like to introduce our speaker for today, Brian Polo, who has been a joy to work with in different capacities over the last few years. The title of the talk, as you can see on the screen, is similar to the paper. Just a short bio about who Brian is. Brian Polo is a research scientist at the Center for Global Health Research at the Kenya Medical Research Institute in Kisumu, Kenya. Brian's research focuses on the molecular and population genomics of malaria vectors, vector surveillance, insecticide monitoring, as well as evaluation of vector control tools. Brian, thank you so much for joining us. You may begin your presentation.
Thank you very much, Pablo. And thanks everyone for purposing to attend this journal club. Before I start my presentation, allow me to acknowledge the wonderful individual listed in my title here for their support and in various levels and in making this work a success. I will be presenting on genomic surveillance, which reveal geographical heterogeneity and differences in known and novel insecticide-resistant mechanism in Anopheles arabiensis across Kenya. For starters, as we all know, the global malaria burden has increased by 33 million between 2019 and 2023. WHO Africa bears the greatest burden of 94% of these cases. Kenya had 3,294,000 cases, 3,294,000 cases, and more than 11,000 deaths were reported. And it was ranked the 14th country in the highest global mortality, as shown in the pie chart on the left of your screen. In the fight against malaria, vector control intervention has demonstrated as a major contributor of malaria reduction. The volcano plot indicates modeling work by Bart et al. Just a minute, which actually indicates that between 2000 and 2015, ITNs and IRS averted a cumulative 79% of malaria cases in Africa. In the recent years, we have seen an increase in evaluation of additional tools to add into the vector control paradigm. Besides the ITN and IRS that are approved by WHO, other tools that are either under partial approval or are still being evaluated include the spatial repellent, use of insecticides, larval source management, use of attractive sugar baits, and also use of transgenic strategies. These vector control tools may be efficacious, but could lead to introduction of selection pressure on the vectors with a non-fitness cost. As we can see in the IR mapper of Africa and Kenya, the red dots indicating confirmed resistance, the yellow partial resistance, and the green susceptibility. Africa is generally marked relatively red, as we can see. And this means that we actually need to do something. So what do we actually need to do? For us to protect our population from malaria, we need to identify working control vector intervention. We also need to understand humans and vector behavior that could prevent interventions from achieving their maximum efficacy. The gap in protection is when our interventions does not overlap with the human behavior and also the vector behavior. What are we currently doing? As I had mentioned earlier, we have continued to introduce new vector control tools to manage this, to manage the vectors. We have also evaluated the human behavior and also tried to influence this behavior for the tools to work optimally. Where it is not feasible to change human behavior, there has been recommendation of addition of tools to complement the already existing ones. The big question is, what do we usually do to the vector to understand its behavior and also to be able to bridge the gap? The continued introduction of vector control intervention in both public health and agricultural pest control has shown to influence selection pressure, hence modifying the behavior of the vector. This study looks to bridge the gap by proposing to incorporate genomic surveillance into routine vector surveillance. Looking at the landscape of malaria in Kenya, the burden of malaria in the country is currently varied. As you can see in the map, high and moderate transmission is recorded around the lake, Lake Victoria, extending to the northwestern part of the country and also at around the coastal part of the country. It is important, generally also within the country, we do observe a prevalence of about 6% and the highest malaria burden of 19% at around the lake region, that is Lake Victoria region, followed by 5% around the coastal region. It is important to mention that even in very low transmission regions, we can observe some malaria hotspots. For example, around Mwea in Kirinyaga County, there have been several cases of malaria. 71% of the country is actually at risk of malaria, and about 15% of outpatient consultations within the country are malaria related. How did we do this work? We started by sampling Anopheles larvae from different sampling locations. And then the larvae were taken to the lab and reared to adult. Adult female mosquitoes that were 3 to 5 days old were exposed to discriminatory doses of insecticides for 30 minutes and observed for up to 72 hours for mortality. Both the resistant and susceptible mosquitoes were sent to Wellcome Sanger Institute for Illumina high-seq whole genome sequencing. Let's now look at the result. We'll start by looking at a bioassay outcome. And here, the map on the left indicates our collection counties. Right at Indian Ocean, around Indian Ocean, we have Kilifi and Kwale County. Here, samples were collected from both Kilifi sub-county and also Kwale sub-county. And in the central of the country, we also did sampling in Kiambu County, where samples were actually collected in Thika sub-county, and in Kirinyaga County, where samples were collected from Mwea. In the western part of the country, we did collection in Busia County, within Teso North and Teso South sub-counties. And we also did some collections in Turkana County around Kakuma. The table indicates our bioassay outcome, where we used two pyrethroids, which is alpha-cypermethrin and deltamethrin, and one organophosphate, that is primifos-methyl, to evaluate mosquito resistance, and this was done using WHO guidelines for bioassay. We then recorded whether mosquitoes were alive 72 hours after exposure, and those were recorded as resistant, and the ones that were dead were recorded as susceptible. Kilifi samples were just included here, but we did not sample them for bioassay. The Kilifi samples included here are part of AG1000 genome that was submitted earlier. Looking at the population structure, we did a principal component analysis using 100,000 biallelic SNP window on a region free of polymorphic inversion, and that is on the chromosome 3L. The PCA revealed two major grouping. The first group was samples from the east of the Rift Valley, which includes Mwea. And so east of the Rift Valley, as you can see in the map, we have Mwea, Thika, Kilifi, and Kwale. And the second group indicates samples that were collected from the west of Rift Valley, and that is Teso and Turkana. We also do note that in 2019, the Turkana collection formed a very separate grouping that spread out quite widely. We also did calculate genetic differentiation statistics, and here the pattern observed here was actually very similar, concurrent to the PCA, and we do observe greater FSTs between samples from the west of the Rift Valley, that is samples from Mwea, Thika, Kilifi, and Kwale, and those samples that were collected from, west of Rift Valley, that is Teso and Turkana, and those samples that were also observed from east of the Rift Valley, which is Mwea, Thika, Kilifi, and Kwale. And that is, as you can see, our FST between Turkana, 2006 sample collection, and Teso 2019, we just have a 0.002 FST difference. But if we do compare Turkana, that is 2006 against samples like, for example, Kilifi on the other side of the Rift Valley, we see a 0.007 difference. And actually it's between 0.5 to 0.005 to 0.007 on the east of the Rift Valley. And as we saw earlier in the PCA scatter plot, individuals collected from Turkana in 2019 formed a somewhat separate dispersed group. Investigation using diversity summary statistics revealed that cohorts from Turkana collected in 2019, as you can see in the bar graph, presented high values of genetic diversity and low Tajimas D, suggesting that this region is subject to populations expansion. Looking at the target site, insecticide resistant, we evaluated the frequency of amino acid mutation at major insecticide binding region, and that is the VGSC, S1, and RDL. Among this target site, only the VGSC L995F substitution, which is associated with resistance to DDT and pyrethroids, was observed at appreciable frequency, as we can see here. And that is only in Turkana. To investigate whether the L99F mutation could explain the observed resistance to pyrethroid in Turkana, difference in SNP allele counts was tested by significance association test with bioassay outcome that is either dead or alive, and we used the Fisher exact test. From this, we did observe that the frequency of L99F was significantly high in alpha cypermethrin, alive mosquitoes from Turkana versus those that were dead. All the other comparisons were not significant. We also did identify, for the first time in Anopheles arabiensis, CNVs at S1, and that is which is a target site for organophosphate and carbamate insecticides at low frequencies of between 4 and 7, and that is particularly in central Kenya and the Mwea region. The presence of L99F mutation in Kenya was interesting, given that it's generally associated with West and Central African population of Anopheles gambi and Colutsai. To understand whether it originated, where it originated from, we explored AG1000 genome phase 3 dataset, but the mutation, as we can see in the heat map, was not detected at all in East Africa. But rather, we saw in Houthi, and this was particularly in Burkina Faso. The only challenge with these samples is that there were only 3 and 1 had the mutation. We were therefore not able to do haplotype comparison to see if there was a selection sweep suggesting that there is sharing of these genetic characteristics. We then assessed for evidence of adoptive integration using haplotype clustering of the VGSC gene on Anopheles arabiensis and Anopheles colutsai from Turkana region, as we can see in the haplotype clustering below. Here, it did indicate that there was no sharing that occurred between these 2 species, ruling out the possibility of adoptive integration. And that is the reason is that you can see the orange samples, which are the colutsai samples from Turkana, clustered very separate from Anopheles arabiensis from the same region. So where did this mutation originate from? It's a good question, but so far from our results, we are not able to answer this. We need to investigate this further. Looking at the metabolic insecticide resistance, here we looked at the frequency of copy number variation at locus previously associated with insecticide resistance, which include the cytochrome P450 gene cluster. We also have carboxylesterase, and that is the carboxylesterase, these particular genes, and also the GST2, that is the glutathione S transferase. The highlighted region indicates fixed resistance or near fixation that we did observe for CYP6 gene clusters and carboxylesterase genes that was very significant for samples collected from central Kenya. For this CNV, we also tested whether the distribution of CNV amplification across the CYP6 gene cluster was associated with the bioassay outcome of whether the mosquitoes were alive or dead using Fisher exact test. And our result observed that a significant association existed between CYP6 gene cluster CNVs and both alpha-cypermethrin and delta-methrin survival in Turkana in particular. We also observed a significant association between CYP6 gene cluster CNV amplification and delta-methrin survival in Thika, and a significant association with primifos methyl in Turkana, Teso, and Thika. Looking at the CYP9K1 CNV, we did observe, and that is this region, we do observe a very high frequency of up to 66%, and that is particularly in Turkana. And the Fisher exact test revealed a significant association of CYP9K1 with mosquitoes from Turkana that survived delta-methrin and alpha-cypermethrin bioassay. This actually do suggest that CYP9K1 could increase pyrethroid resistance in and around Turkana region. We also did observe high CNV amplification of up to 100 at carboxylesterase, and that is the COEF2, COE2F, and also the COE gene that is between 2G to 7G. Of all these genes, we observed very high frequencies, and that is particularly in central Kenya compared to the rest of the country. And looking at this actually, I would also want to mention that this specific gene is also associated with resistance to organophosphate, that is particularly primifos methyl, and amplification at this loci was significantly associated with delta-methrin and primifos methyl survival, and that is in central Kenya. On the other hand, that is the COE2F amplification was associated with alpha-cypermethrin survival in Turkana. We investigated the presence of novel candidate gene conferring resistance, and that is using genome-wide selection scan, H12 heterozygosity statistics. As expected, signals of selections were observed in non-resistant loci. As you can see, that is the CYP6 gene. We did have signals of selection. We also observed selection peak at KIP1, and this is particularly in samples that were collected from central Kenya. And here, what we are showing are just samples that were collected from central Kenya. Beside CYP6, we also did see carboxylesterase, peaks at carboxylesterase gene, but we did observe a peak at KIP1 gene for samples that were collected from central Kenya. The KIP1 gene has been associated with regulation of transcription factor, which is known to trigger the expression of multiple metabolic resistant genes, including cytochrome P450 and glutathione S transferase in response to oxidative stress, and that was reported in Anopheles, Gambia. Since no particular variant has been associated with KIP1 gene, we did a further investigation using diplotype clustering to assess whether either CNV duplication or SNP was associated with the candidate diplotypes under selection. And in particular, we had 3 diplotypes in central Kenya. Of all the 3, we did observe one very unique one that had the 2 SNPs, that is the E762 and the D780N. These two were unique because within them, the E762 was a stop-gain mutation, indicating a potential loss of function. The role of KIP1 gene within the transcription pathway is to act as a repressor that prevents the unnecessary expression of a detoxification gene in the absence of stress. It is possible that the loss of function that we are observing could result in elevated antioxidant effect. We therefore do recommend that further studies on this gene is actually still required. On the discussion, we do have the first key question is, what is driving resistance that we are observing in Kenya? Could it be geographical isolation or selection pressure? As had been mentioned earlier, the PCA and FST did reveal two grouping, and that is, as we can see, the map of Kenya indicating a proximal position of the Great Rift Valley. The PCA did indicate that samples from west of the Great Rift Valley, that is Teso and Turkana, were separated from samples from the east of the Great Rift Valley, that is Mwea and Thika. They observed high heterogeneity in population, genetic structure, and diverse insecticide resistant mechanism across the country is likely a driver of restricted gene flow, and that is possibly due to the presence of the Great Rift Valley. Does that rule out selection pressure? Not at all. In particular, I did highlight very high organophosphate resistance from our results in central Kenya. And particularly in Mwea, we have actually had reports of very large-scale rice farming in Mwea. And also keen to note is that Mwea in particular is categorized as a low-risk area with less than 1%. So that is central Kenya, where Mwea is categorized as a low-risk area with less than 1% prevalence. The only vector control intervention, the main vector control intervention that is actually administered in this region is standard pyrethroid nets, and that rules out the possibility of vector control contributing to the resistance that we see in the mosquitoes collected from that region. The highest possibilities that the organophosphate resistance could be attributed to the agricultural activity in Mwea. Similarly, in Thika, there is very large-scale horticultural farming where they also use agricultural pesticides, which could contribute to the results that we saw. Another region within the same country, that is the Turkana region, is home to one of the largest refugee camps, and that is Kakuma, with a population of about 290,000 people. And this region is known for seasonal malaria outbreak, that is between May and October. Last year, around the high peak, malaria cases were recorded up to 850 per week. And ideally, with such very high cases, it could prompt deployment of various vector control intervention. In particular, in the region, the most used vector control method is deployment of pyrethroid ITNs intervention, which is coupled with other chemotherapy preventions. We could therefore attribute the resistant profile we see which is the VGSCL95F mutation and the CYP9K1 duplication, which are mainly associated with pyrethroid and DDT resistant to the likely emergence of intense selection pressure in the region, where high population density has necessitated the widespread deployment of pyrethroid-treated nets, creating a resistance hotspot. In conclusion, it would be paramount to mention that region-specific tailored malaria vector control strategies are very important within the country, as I had highlighted, because of the various profiles of resistance in the different parts of the country, other than just using a one-size-fits-all approach. This study also brings out the importance of incorporating genomic surveillance into our routine national malaria control programs. And then it would be important also to do whole genome sequencing on samples that are routinely collected for entomological surveillance. The study also do recommend that it is necessary to do genomic analysis, especially during interventions which will also help identify insecticide resistance that might compromise the effectiveness of the new tools that are being introduced. And it could also identify novel insecticide resistant mechanisms that are introduced by the tools. The study also do bring out the probable importance of KIP1 gene, which should also be evaluated further to understand its insecticide resistance, especially as we can see in central Kenya. I'd like to acknowledge in particular Dr. Luna Kamau, who had the funding for this and led this activity. Dr. Eric Ochomo, who is my supervisor, together with the rest of the team from KEMRI, Kelly in particular, who assisted a lot in both the analysis and also bringing out the manuscript, together with Alistair. I also do appreciate Charles Mbogo, Elijah and Sonia from PAMCA, David Mburu from Pwani University, and also Edith from Division of National Malaria Control Program, and the rest of the team who I wasn't able to mention here, and also Malaria Gen for both the training and also the supporting analysis. Thank you very much, and I hand it back to Pabalo.
Okay, thank you so much, Brian. That was fantastic. Well-paced, well-explained. I think you were very calm, so well done, and thank you so much for your time and presentation. We've now reached Q&A time as well as comments. So please do raise your hand. We'll allow you to speak, or type your question in the Q&A and chat box, and I'll read them out for Brian. We have about 25 minutes to go, so please do engage. But yeah, also follow Brian on LinkedIn, connect, if you want to collaborate or have further questions as well. But for now, we do have about 24 minutes, as I say, for Q&A. I'll start while other people are still thinking about their questions. I think I have a lot, but I'll try. Maybe let me start with one. Let me not be too excited as well. So I think in the beginning, in your first slide, you showed the data that between 2019 and 2023, there was an increase in malaria cases throughout the world, but particularly in Africa, and then also in Kenya. So malaria cases, as well as deaths, increased across Africa, but particularly in Kenya. And I think you ranked quite highly there. So my question is, what do you attribute the increase in malaria cases and deaths between that period? Would COVID be a factor? Maybe governments focused more on fighting COVID in 2020, 2021, 2022? Or are there other factors that you outlined in your presentation, such as resistance, etc., that could contribute to the increase in malaria cases and deaths between 2019 and 2023?
Thank you, Pabalo, for the question. I'll start by mentioning that Yes, COVID-19 could be a key factor of this change, especially the current increase of about 34 million. But then you know that malaria is mainly transmitted by a vector, and that is Anopheles mosquitoes. And any chance that we offer to them to proliferate, to continue proliferating even within our environment, two things happens is that it continues to interact with our environment, that is both the chemicals that we introduce, and also it changes its behavior. So I would say that COVID-19 did a big, contributed greatly, especially in Africa. But then on the other hand, the control interventions that are being introduced currently also needs to be beefed up, because probably is that around that time, a lot of attention was more into COVID-19.
Thank you so much for that answer, Brian. The floor is open. Kindly raise your hand or type your questions in the Q&A, and I'll read them out for Brian. I think I also might have seen Kelly in the room. Kelly, if you have anything to add, please kindly raise your hand as well. I'm putting you on the spot, apologies. But if you wanted to say anything else, please do kindly also add your contributions to the discussion. Whilst people are thinking, I don't know if you, Brian, there was a paper by Yao Badiako from Ghana. I think he's the CEO for a science institute that side, who wrote a paper last year in Cell on the interaction between malaria and COVID specifically. So they're claiming in that paper that the reason why, particularly in African regions, there was a sort of low COVID rate compared to the rest of the world was due to people having had malaria before. And I found that quite interesting as well to say there is an interaction genetically between malaria and COVID as well.
That's interesting. I would love to read it, especially.
I'll try to find the paper in the meantime while other people are asking questions as well. So yes, as I say, audience, please do raise your hand or type your questions in the Q&A. And Brian will gladly address your questions. Okay, I think there's a question.
It looks like Kelly has her hand up.
Okay, wait. So I was gonna thanks, John. I'm seeing there's two questions, so I'm excited while I was trying to find the paper. So I'm gonna give Kelly a chance to say something, and then I'll read the questions that I'm seeing in the comments. Kelly, go ahead. Kelly?
Hello. Yeah, I was waiting for the message to come up to say that I wasn't muted. Sorry. Yeah, no, I just wanted to say this is fantastic work from Brian, and I think that these findings really highlight that even in a single Anopheles species, you get such a variation in insecticide resistance mechanisms across a geographical area, and so it just sort of highlights the need for more finer scale genomic surveillance studies. So that more targeted malaria interventions can be applied. So yeah, I think this is a fantastic example of that. And yeah, thanks so much to Brian for his hard work.
Thanks, Kelly. Thanks for your contribution too.
Thanks, Kelly. There's a question from Messi Bella Omunagbe. Do you think the KEAP1 gene can be considered as a single genetic marker for determining insecticide resistance rather than checking all those many SIP genes That's from Messi. Brian?
That's a good question. I'll move to keep one gene, and as I'm moving there, I would like to mention that it was interesting to find keep one gene, particularly in one region within the country. And her question is whether it could be targeted on a specific SIP other than looking at the very many SNPs that I did present. Yes, it's a possibility. But one thing is that we have never actually attributed KIP1 gene to any variation, whether SNP or SIP, the CNVs or SNP variation. And for that reason, we need to check through as much as we can to identify which could be a driver to possible resistance. And that is the reason why I did diplotype clustering. And in particular, the fact that E762 is a stop gain mutation, which indicates that there's a possible loss of function, gives us a chance of probably attributing it to association with insecticide resistance. But this area still needs further research.
Okay. Thank you so much, Brian, for that answer. As I say, we have about 17 minutes left for Q&A or comments. So please do raise your hand or type your questions in the chat. I'm going to give it one more minute before I let everyone go, if there are no questions. But as I said, please do follow Brian on LinkedIn and also email him if you have any further questions regarding the presentation as well. And as I said, the presentation has been recorded and will be available on the Malaria Gen channel later today, so you can share it with your networks as well. Going once, going twice. Okay. Brian, before I let people go, do you have any last comments from your side?
None in particular. I'd like to appreciate you for considering me to present in this journal club, and thanks for the consideration.
Thank you so much, Brian. It was a brilliant presentation. Thank you so much for your time and educating us on malaria in Kenya. It was really enjoyable and educational as also really appreciated. And to all of you participants, thank you so much for joining us. Please sign up to the Malaria Gen newsletter to receive announcements about future events as well. We'll have another journal club sometime in May, rather in April, and we will send out announcements via email and also on our social media sites on LinkedIn X, as well as Blue Sky. So please be on the lookout for that. But in the meantime, as I said, please continue engaging in our content and in the lessons that we learned today. Thank you so much, everyone. Have a good day and see you next month. Goodbye.